Welcome to Biotic World. Today we are exploring one of the most fascinating processes in molecular biology, DNA transcription. It is the first process in gene expression where the genetic code in DNA is converted into a readable masses, paving the way for protein synthesis. Let's break it down step by step. What is DNA transcription? DNA transcription is the process by which a specific segment of DNA is copied into RNA, particularly messenger RNA or mRNA. This happens in the cell's nucleus, and it's a crucial step in translating genetic information into proteins. But why is this necessary? DNA stays protected in the nucleus, so mRNA acts as a temporary copy or a messenger carrying instructions to the ribosomes in the cytoplasm. So there are the several key players in the DNA transcription. Let's have a look. Before we dive into the process, let's meet the key players. First, the DNA template strand, the blueprint from which RNA is synthesized. Next is RNA polymerase, the enzyme that reads the DNA and builds the RNA molecule. Next is the nucleotides. These are the building blocks of RNA like the adenine, uracil, cytosine, and guanine. Fourth is the transcription factors, proteins that help initiate and regulate transcription. Next, we will have a look to the steps of DNA transcription. Transcription is divided into the three main stages, which are the initiation, elongation, and the termination. First step is the initiation. Transcription begins when transcription factors bind to a specific sequence on the DNA called the promoter region. The most common promoter in eukaryotic cells is the data box. DNA polymerase then attaches to the promoter and binds the DNA helix near the gene to expose the template strand. Our second step is the elongation. Once bound, RNA polymerase moves along the template strand in the 3' to 5' direction, synthesizing RNA in the 5' to 3' direction. Complementary RNA nucleotides pair with the DNA template, where A pairs with U and C pairs with G. The RNA strands elongates, growing as a single strand. Our third step is the termination. Transcription continues until RNA polymerase reaches a terminator sequence on the DNA. At this point, RNA polymerase releases the newly formed RNA strand, which is now called the pre-mRNA in eukaryotes. Our fourth step here after the transcription, that is post-transcriptional modifications in eukaryotes only. In eukaryotic cells, the pre-mRNA undergoes several modifications before it's ready to leave the nucleus. These modifications are capping, polyadenylation, and splicing. So let's talk about the capping. A 5' cap is added to protect the RNA from liquidation and assist in ribosome binding. Second is the polyadenylation. A tail of adenine nucleotides called the poly A is added to the 3' end of the for stability. Next is splicing. Introns, the non-coding regions, are removed, and hexons, the coding regions, are joined together to form a mature mRNA molecule. Now let's talk why transcription is important. DNA transcription is vital because it's the first step in expressing genes. Without it, cell could not make the proteins necessary for structure, function, and regulation. It's how the information stored in your DNA gets turned into action. Here are a few fascinating facts. Every single cell in your body contains around 2 meters of DNA, yet transcription happens with astonishing precision. In bacteria, transcription and translation can happen simultaneously, unlike in eukaryotes. Errors in transcription are rare but can lead to diseases if they occur in crucial genes. Here is the conclusion. That's DNA transcription, a process that's fundamental to life as we know it. By understanding how genes are expressed, we can unlock secrets of biology, medicine, and even evolution. Thanks for watching Biotic World. If you found this video helpful, don't forget to like, share, and subscribe for more fascinating biology content.